Hi everyone, welcome back to my channel. Today I would like to move on to the root developments. Roots development is one of my favorite subject because the development of roots to give different function is very specific to different species, and it contains a lot of variations between species. So let's look at the function of the roots. So when penetrate to soil, which is often hard and potentially damaged to a growing root via frictions, it absorb water and dissolve mineral from the soil. So the root also will act as a filter to substance absorbers to exclude undesirable compounds such as the salt from the water. It can say that some of the maybe toxic compound can be also excluded, but recent findings show that the microplastic, which is a small size of plastic that degraded from the plastic material, can be absorbed into the plant cell. So it is now become a major concern that the accumulations of this microplastic in plant cell can be then transferred to the human or animals sort of when we consume the plant cell. So how plant exclude this microplastic and whether it will accumulate in the fruits or seed of the plant, which will be later consumed by the human, is still in a very new research subject. So a part of the act as a filter is we transfer water from the roots to the shoot of the plant. So transport photosynthesis such as carbohydrate produced by photosynthesis from shoot to the root, where is distribute to support the respirations and growth of the plant. So some of the roots also will serve as a storage organ for photosynthesis. So we know that most of the roots serve as an anchor and support to the above ground portions of the plants. So all these functions are accomplished by specialized tissue with specific physiological function. So this is two major root types. So Dicot have a taproot system. So while monocot has a fibrous root system, which is also known as adventitious root system. A taproot system has a main root that grows down vertically. If you can see, this is the main root, and it's go down vertically, from which many smaller lateral shoots arise. So you have many lateral roots grow out from the main root. Dandelions are a common example. The taproot usually break off when these weeds are pulled from the ground. They can regrow another shoot from the remaining roots. That is why, for example, glasses, when you didn't pull it out properly from the ground and you just chop off the shoot, it can be very fast regenerate the whole plant again. A taproot system penetrate deep into the soil. In contrast, a fibrous root system is located closer to the soil surface. So this is a fibrous root, and because it's grow not deep down to the soil, but it have a lot of the small lateral roots. The fibrous root system is located closer to the soil surface, where it forms a dense network of roots that also help prevent soil erosion, such as the grasses as a good example, and wheat, rice, and corn. Once the plant is developed completely, the fibrous root appear as a mat underneath the plant. The root in the fibrous root system move more horizontally than vertically, but they cannot penetrate deeper into the soil. So the fibrous roots are considered as surface feeder, as they do not penetrate deep into the soil but feed on the surface soil and organic matter. So organic matter means that the, for example, leaf that fall down from the plant, and it start degrade and providing organic matter. So fibrous root also cannot act as an organ for food storage like in some plant with the taproot system. So like carrot is a taproot system. Because they are more fixed on the surface of the soil, the fibrous roots is considered crucial for the prevention of soil erosion as they hold the surface soil firmly. Some plants have combinations of taproots and fibrous root. So plants that grow in dry area often have deep root system, whereas plants that grow in area with abundant water are likely to have shallower root system. So this diagram shows the primary growth of the roots. Root tissue are produced from root apical meristem by a highly stereotypical pattern of cell divisions. Root meristem resemble shoot meristem but have two important differences. So the first is they have a root cap cover the root meristem. So this one is a root cap that cover the meristem cell, and no segmental arrangement as seen with the node internode module of the shoot. So the root is set up early in the late hatch set embryo but a set of initial cell. So you can have to recall this is apical cell, this is a basal cell, and is developed from when during early globular stage. So each column of root cell originate with a specific cell in the meristem via a specific pattern of cell divisions. The color code show the different cell type. So these hypotheses will develop into different cell type. So this different cell type, the cell type here and here and here is different depending on the color code. You can see that is a different cell type in the root system. So let's look at the meristem region. So this here are the very tip of the apical meristem is the quiescent center. The rate of cell division is much slower than daughter cell of the apical meristem around them. So this structure is located just behind the root cap and consists of 500 to 1000 seemingly inactive cells. So this is the quiescent center and the root cap is here. So what is the function of this quiescent center? So these cells are usually in the G1 phase of the cell cycle and divide only about once every 15 to 20 days. Division of initial can be either solely antiquinal, which is orthogonal to the axis of the growth, resulting in a single file of cell or first antiquinal, then periclinal, which is the parallel to the axis of the growth. So it can be upwards or horizontal like that. This will result in two or more cell layer. So the columella initial generally divide only anticlinally, and their progeny undergo rapid cell expansion and then differentiate, producing starch containing amylophase that play a role in gravity sensing. The other three types of initial generally undergo both anticlinal and periclinal divisions, resulting in cell lineage that acquire different identification. For example, the cortex or endodermal initial divide first anticlinally to regenerate the initial cell and basal daughter cell. The basal daughter then undergo periclinal divisions to form the first cell of the cortex and endodermal linkage. This cell will undergo a smaller number of anticlinal divisions, during which time they will acquire their specific fate. Subsequently, they will undergo rapid polar expansion. The cell in the Ginseng center function as a reservoir to replace damaged cell of meristem. It is important because it organizes the pattern of primary growth in root. To the outside, the cell of the root cap are formed. So this one to the outside. Large parenchymal cell which protect the apical meristem as the root grow through the soil, constantly being worn away and replaced. So this is a reservoir because it will keep maintaining the cell number instead of rapidly divided.
So look at this picture. Anyone know what plant is this? This is a wild ginseng. So you can see it's actually propagate via seed germination. So the wild ginseng is grown naturally in mountain, and mostly it's a more cooler area, and it's handpicked by the gatherers and sold it in the market. So the wild ginseng plant is now almost extinct in China and endangered globally. This is due to the high demand for the product in recent years, lead to the harvesting of wild plant faster that they can grow and reproduce. So when you look at this wild ginseng, you start to think, what is the medical value of this plant? So it's actually the medical value is on their roots. So this picture shows that the edge of the ginseng roots that in between 10 to 35 years old. So the sizes here is range from 1 to 3 inch long. So what is the prices of this this ginseng? The ginseng is actually price range by the size and also of course by the age. Some of the ginseng can be as old as 10 and above 100 years old. So for the range between 10 to 35 years old, the prices is around $1,000 US per pound. So we know that 1 pound is equivalent to 0.45 kg. So the commercial value is due to the good medical value from this ginseng. Is actually consumed, and Chinese belief that is contribute to the longevity. So because it is highly demand and almost endangered, so scientists use the knowledge in tissue culture and the roots development to culture this ginseng. So this picture show that the root tissue culture of the ginseng. So you can see that the meristem cell of the root tissue being cultured in the tissue culture in the accepted conditions using the MS media, and then after that you can micropropagate. That means you can propagate the meristem root tissue in bulk cultures, and then you can each of these bottles you can maximize in a bioreactor, and this one is the end results of the ginseng. Of course, the age of the ginseng also contribute to the their medical value, but now it's proof that the tissue culture can produce a big amount of these ginseng roots in a short time. Plant root zone has been divided into 3 regions: the zone of cellular divisions, cellular elongation, and cellular maturation. So root growth begin with seed germinations. When the plant embryo emerge from the seed, the radicle of the embryo form the root system. The tips of the root is protected by the root caps structures exclusive to roots and unlike any other plant structure. The root cap is continuously replaced because it is easily damaged as the root push through soil. The root tips can be divided into 3 zones: a zone of divisions, a zone of elongation, and a zone of maturation. The zone of cell division is closest to the root tips and is made up of activity dividing cell of the root meristem. So we call it as also meristem zone, which contain the undifferentiated cell of a germinating plant. The zone of elongation here is move outwards, and zone of maturations is here. The zone of elongation is where the newly formed cell increase in length, thereby lengthening the shoot. Beginning of the first root hair is the zone of cell maturation. You can see here where the root cell differentiates into specialized cell type. All 3 zones are approximately the first centimeters of the root tips. So let's look at the zone of cellular divisions. It's around the quiescent center and is a dome-shaped apical meristem located 1.5 to 1.5 millimeter behind the root tips. Yeah. The meristemic region of the zone of cellular divisions and is made of small density cytoplasm cell. Meristem cell in root divide every 12 to 36 hours. In some plant, the meristem produce almost 20,000 new cells each day. So you can see in the cross sections here, the protoderm will be here is the protoderm will be developed into epidermis. Ground meristem is a cortex, then including endodermis. So cortex here, endodermis is the green cell here, and procambium is a pericycle, primary phylum, vascular cambium, and primary xylem, and the pitch is inside in the center of the zone of cellular division. So which this one is a procambium, is the center. So this one is a center. Let's look at the elongation part. So the elongation area occur 4 to 15 millimeters behind the root tips. So it's here. Cell in this zone elongate by as much as 150-fold by filling the vacuole with water. This zone is easily distinguished from the root cap and zone of cellular divisions by its long vacuolate cell. So this cellular division elongation in the elongating zone through the root cap and apical meristem through the soil at rate as high as 4 cm per day. Cells behind the elongation zone do not elongate. They are in maturation zone. So this is maturation zone. Most of the cell mature or differentiate into various distinct cell type of the primary tissue, occur 1 to 5 cm behind the root tip, sometimes called the region of differentiation or root hair zone, because it's only the maturation contain the root hair. So root hair increases the absorptive surface area of root several thousand-fold and are usually less than a millimeter long. In most plants, they form from asymmetric divisions of the protoderm and usually live only a few days. with all hair further from the tip constantly being replaced by new one closer to the tip. Root hair only form in maturing, non-elongating regions of the root. Because the root hair are fragile extensions of epidermal cell, then they usually break off when plants are transplanted. So this picture show the differences of dicot root and monocot root. So as we know that the most of the monocot root, they doesn't have this secondary growth, so only dicot roots can have or can develop in a secondary stage. So we can see that the structures of cortex ground tissue is quite different in dicot compared to monocot, and also the vascular system here is like a cross, but the vascular system in monocot is more scattered around. And this table show there is 5 typical characters differences in dicot and monocot roots that you need to familiar. And let's look how the lateral roots develop from the taproot system. So the lateral roots primordia arise from pericycle cell opposite xylem pole at some distance from the primary root meristem. So this one is pericycle cell and is actually emerge or grow out from the pericycle cell. The initial cell divisions pattern that give rise to new primordia are very different from those occurring during primary root formation. At later stages of lateral root formation, the cellular organism become very similar to that of primary root. Although lateral roots display more variability in cell number and precise cellular organizations, analysis of cell divisions pattern aided by enhancer trap in that marks various root tissue have been used to stage lateral roots development. X stages were defined based on specific anatomical characteristics and cell divisions. Expressions from cell type specific marker line was used to identify the cell type in the developing primordium. Genes expression analysis of the expressions pattern and cell divisions pattern reveal that the remarkable amount of organization and cell differentiation occur at every early stages of lateral roots primordium development, with differential gene expressions appear after the first set of division of the pericycle. In contrast, the lateral root meristem does not become active until after the primordium emerges from the parent root, and therefore does not appear to play a role in early pattern formation and organizations. So this one is pre-emergence, emergence, and then meristem activation in lateral roots organizations. So if you can see, this one is a cross sections of the root. This one is the pericycle and is emerge actually from the pericycle. So you see this one lateral roots arising from meristem activity of a pericycle, and if you push the cell and then come out as a lateral roots. Well, adventitious shoots is a root initiate at site other than the root pole of the embryo, is formed naturally in many plants. Cutting of stem or leaf picture and by layering can also initiate or activate the formation of adventitious roots. Arise from the stem or leaf and not from another roots usually occur in monocot and peridophytes. Few only happen in dicots. Initiated by cell divisions in the deeper lying parenchyma cell associated with vascular tissue. Stem and leaf of seed plant generally do not have pericycle, and a root primordium is organized similarly as in lateral roots formation and then grow out as a new root. So that is the adventitious formation. And here you can see that is a lot of the different types of root structure. So root structure may be modified for specific purpose. For example, some roots are bulbous and store starch, such as this, and aerial roots and pops root are two forms of above ground root and provide additional support to anchor the plant. Tap roots such as the carrot, turnip and beets are examples of roots that are modified for food storage. So many vegetables are modified roots. Epiphytic roots, this one is epiphytic roots, enable a plant to grow on another plant. For example, the epiphytic roots of orchid develop spongy tissue to absorb moisture. So it's actually the root of orchid very sensitive to water. That's why you are not advised to grow the orchid inside to the soil or some container that can retain water, because that will damage the root. So the root of the orchid was saying that have lung, is a lung system of the orchid, which they can absorb moisture, and that is the water supply for the whole plant. The biome tree, which one is this one is the biome tree, begins as an epiphytus germinating in the branches of the host tree. An aerial root develop from the branches and eventually reach the ground. You can see the roots develop from the branches, and then it's providing additional support for the plant. In skew pine, a palm-like tree that grow in sandy tropical soil, above ground pop roots develop from the nodules to provide additional support. The biome tree, that's also known as a straddler fig, begin life as an epiphytic in a host tree. Vegetative roots is for propagation, so you can actually isolate the plant here and transfer to grow to a new place, and it will start to propagate. Okay, so now look at the modified root. The first picture here shows the parasite root. The Haustorium roots penetrate the tissue of a host cell and absorb nutrient and water from its host. So this parasite root system can be found such as in the dodder plant. So the Haustorium form of a vascular union with the host plant to redirect the host nutrient. And the second one here show the buttress root that are very large, wide root on all sides of a shadowy rooted tree. Typically, they are found in nutrient poor tropical forest soil that may not be very deep, so they prevent the tree from falling over while also gathering more nutrient. contradictile roots continuously pull the plant deeper into the ground as stem elongates, so it can remain subterranean or at an appropriate level in the ground. The example is the lily bud. Somatophore, that's always found in the mangrove area or swamp pit, is an aerial root specialized for gaseous exchange. And then the final is the mycorrhiza roots. So mycorrhiza roots is the form of the root together with the fungus. So the function is to exchange the nutrient from the is a kind of the symbiosis relationship of the fungus mycorrhiza roots. So now we move on to how the secondary growth of the root in dicot plant. So the secondary growth occur in many roots and usually result in the thickening of the root diameters. So means that is make this diameter more thick by additional vascular tissue. So initiations of secondary growth occur when the cell in the residual procambium and part of the pericycle begin to make periclinal divisions. So this is this area that you can see they start to propagate. Only the pericycle cell opposite the xylem point start to make periclinal divisions. The inner layer of the cell become the vascular cambium. The outer layer is retained as pericycle. The vascular cambium is continued around the primary xylem. So if you continue, the vascular cambium will continue divide periclinally, and the daughter cell that result from this division differentiate into secondary xylem cell if they divide off towards the inside of the root, or secondary phylum cell if they divided towards the outer surface of the root. After many cell divisions and cell differentiation, the root exhibiting secondary growth look like this. And then the pericycle resume its meristemic character and begin divide periclinally again. So this one is the pericycle derivative. At this point, it is called the phylogens or the cock cambium. So this one is the cock cambium and is developed to call phylogen. The cock cambium form phylum cell is a cock cell towards the outside of the plant. These cells are dead at maturity. They are sub-recite, which make the cell impermeable to water. The phylum cell in cross sections appear in neat order files. The cock cambium also produce the phylloderm, a tissue consisting of cells that are living at maturity. So the locations of the periodium can be seen in the diagram here. So not all the plants follow the secondary growth in their root in this way, and they may not confirm the development pattern that exactly the same as mentioning here. So I think that's all. Here I would like to end my video with showing you the root system that grow together as a part of the ancient Angkor Wat temple in Cambodia. Okay, thank you. I see you guys again for the next chapter.